When my students start trying to study linked genes, initially they get kind of confused, but after you get past that first confusion and you start to learn what it really is, it's actually a pretty simple concept. The Human Genome Project has determined that humans have somewhere in the range of about 25,000 genes. Now, if we only have 23 different kinds of chromosomes, that tells you, if you do the same simple math, that we have roughly 1,000 genes, plus or minus a bit, on average, per chromosome. So linked genes is simply the idea that if you have two genes on the same chromosome, when that chromosome goes into a sperm or an egg, those two genes travel together and wind up being inherited together. So they are, quote, linked. This would violate Mendel's second law, the law of independent assortment. But luckily, there's this process during meiosis, during the first step called prophase 1, that does this process called crossing over, where physically a chromosome will break and exchange parts with its homologous pair, so that even though two genes were right next to each other, maybe they get swapped over. So your mom's version of a particular allele will get swapped over to the DNA that originally came from your dad, and vice versa, so that all of your children don't look the same. Now, one thing about this is that genes that are far apart on the chromosome, they tend to get crossed over very easily because you have lots of room for those breaks and exchanges to occur. But if they're right smack dab next to each other, it's very unlikely that you'll get that crossing over event between them, so they tend to be inherited together. A lot of genetic tests where they test your DNA to see if you have a particular disease or a chance of a particular disease, they're not actually testing for that gene, they're actually testing for a linked gene that they've found tends to be passed on along with the disease, because they're still trying to figure out where exactly the disease gene is. So let's take a look at this and see how it works. Now, here's a standard Mendelian example. Here we have somebody who's homozygous recessive for two genes, the R gene and the E gene. I don't really care in this context what they stand for. And this person is having a crossing event with somebody who is big R, big E, little e, heterozygous for both traits. Now, this person can only create one possible kind of gamete, a gamete, a sperm or egg, that has little r and little e in it. This person, on the other hand, through the process of meiosis, can create four possible gametes. For example, here's their chromosomes with the big R's on the the R gene is on the larger chromosomes, the E gene is on the smaller chromosomes. And if they are aligned this way, when these separate, we wind up getting our R's and our E's like this. Now, that was with the big R's and big E's together on one side. What if they work the other way? What if the big E happens to be on that side, on the opposite side from the big R? What that means happens is that when these separate, I wind up creating a different possible combination than with the earlier combo. So what I'm going to do is I'm going to create for you here the four possible gametes that this one individual could create. So they could make the big R, little e, that we see right here. They could see the big R, big E version. That's right here. We could create a little r, little e, right here. And our last one, our little r, big E, right there. And so when we do this cross, when we have these two individuals have their offspring, we would expect 25% would be this way, 25, 25, and 25. So we'd have equal distributions of our genotypes and phenotypes. Make sense? All right. Let's take a look at what happens when they're not on different chromosomes, when instead the R and the E are on the same chromosome. So here I have, again, the same individual, same genotype, but now the little r and little e are on physically the same piece of paper or DNA molecule. Similarly over here, we have them together on the same DNA molecule. So now when they pair up, yes, the red one could be on my left or on the right, or it can swap around. It doesn't matter. However, they wind up only creating two kinds of gametes, what we see here. All right? So we have only our big R, big E gametes being formed. These guys here, absolutely none of the big R, little e's. There. We have some little r littlies right here, but none of our little r biggies. So 50% of our offspring will be this way, 50% that way. Now, what's that crossing over stuff? Well, let's put them back. You recall from meiosis during prophase 1, the homologous chromosomes come together, and then randomly they get broken by enzymes, and then other enzymes, specifically the enzyme used in your cells is an enzyme called ligase. In this case, I call it the enzyme called tape. And so they cross over like that, and we wind up getting this being formed, and this being formed, and this being formed, and my tape is running out. Too bad. So sad. This being formed. But wait, even though these two chromosomes are now different from those, the crossing over didn't occur between my R's and my E's. So my eventual outcome winds up being the same. And that's because these guys are so close to each other. What if they were further apart? Well, let's take a look at that. Here I have, again, two chromosomes. Again, the R's and the E's are together on the same chromosome, but now there's much further distance between them, which gives, if I make the break at roughly the same spot, there's a greater chance that the crossing over can occur. So now I make the break at roughly the same spot, and now when I separate them, I wind up going back to having something approaching a quarter, a quarter, a quarter. So now I do start to see these sorts of offspring when I do the breeding. Now, it may not be 25, 25, 25, 25%. It may be 15% and 15% of these, and then, let's see, 40, or sorry, let me do my math, 35 and 35% there. But again, we've delinked them because of this crossing over event. So that's linked genes.